Okay.
Oh, it didn't make
It didn't make a sound. Okay. Well, I'm sure that's all right. Okay, cool. Hi, everyone. Welcome to the Skills Seminar. I'm going to do my brief intro for Vince. So this is Vince Buffalo. He is a postdoc in the Nielsen lab. He earned his PhD at UC Davis, working with Graham Coop. I just realized that it's really weird to be facing that way. And then he also did, where he studied population biology. He also did a postdoc in the Kern Ralph collab at the University of Oregon. And he also has worked as a bioinformatician, where he wrote a book called Bioinformatics Data Skills, so very relevant to the seminar. And currently he works on understanding how natural selection can be detected and quantified using genomic data. He overall develops population and genetics, population and quantitative genetics theory, statistical methods, and computational tools to understand how selection shapes genome-wide patterns of variation, as well as diversity across species. Outside the lab, Vince is really into running and fly fishing. And as I recently learned, he's also a big aviation nerd. So welcome, Vince. Good to have you here. So excited to hear about SciDataFlow.
Yeah, thank you, Monica, for that kind introduction. I'm happy to present SciDataFlow, which is sort of a side project that I did at the beginning of this year. Is that right? Yeah. So hopefully you find it interesting. I like the idea that you have these skills seminars, and I've been procrastinating doing one. Sorry, Monica, but I'm happy to give others in the future on other topics if that's a great thing. Eat pizza, learn about comp bio. What could be better, really? So just a quick overview. I'm going to discuss sort of the problem that SciDataFlow is addressing, and then a little bit about the architecture, just because I like to nerd out on some of the details, and then I'll sort of give a walkthrough on how to use SciDataFlow in your own sort of computational biology projects. So what is the problem? What does this sort of solve? Well, let's start with a poll here. How many of you have seen email authors for data in a published article? Raise your hands. Okay. Actually, pretty much everyone. Okay. How many of you have emailed the author to do this? Okay. How many have received no reply? 2. Okay, that's probably like a quarter of the people. How many have seen links to data on an author's personal website in an article? You know, see this, blah, 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 and then some institution from a decade ago that the website's dead. Have you tried to access those links, but they're dead? Has anyone ever done that? Yeah. So this is a huge problem in science. I don't need to tell any of you that this is a giant problem. And it's frankly unacceptable in my view, which is why I wrote SciDataFlow. So the problem is that data sharing in science sucks currently. We don't do it right. This is the public's money. This stuff should be in the public, minus the exceptions for privacy concerns, but that's really a small proportion of total data produced by science. So there's this paper that came out, I think this was last year, that was looking at this, and it's kind of a brilliant experiment. What do? You email authors from various articles and different subjects, and you say, hey, I saw you said, you know, you could provide the data. Can you send me the data? And it was sort of a standard email they sent out to, I don't know what the sample size was, probably hundreds of folks. So looking at ecology, this is kind of shocking in my view, that some, let's say 5 to 6% decline to share the data outright, which is shocking for ecology. There's no really big privacy concerns in ecology. And then I think some, what is it, 65% just ignored the request. The data is gone, who knows where. And then about 30% receive the data. So you're looking at less than a third of these requests actually led to, you know, the person that either wanted to replicate these findings or expand on these findings actually being able to access the data. So looking across other fields, you can see that ecology is bad, I would say, but social sciences are far worse. I think this is really shocking that some 55% are just declined immediately in the social sciences. So I tweeted this figure yesterday. Someone replied and they said the most bizarre reply that I ever got when requesting data was, tell me your idea and I'll test it for you in our data, which is again, unacceptable. That's not how science should work. So yeah, pointing this out here. There's also sort of a decay rate in data availability with time, which makes sense. There's some sort of natural decay in the data due to the data getting lost or people just not really remembering that study or, you know, a variety of different reasons. So again, this is a problem. We know that in science, very few findings are actually replicated or attempted to be replicated. And even if it were necessary to replicate some findings, you wouldn't be able to do it in a large proportion of cases. So this is all sort of the impetus for SciDataFlow. Sociologist Robert Merton sort of came up with these famous Mertonian norms of science. This is the norm of communism, which I think is a bit of a misnomer, but it's sort of this idea that, we should think about sharing our goods. There's no private scientific property. The institutional conception of science as part of the public domain is linked with the imperative for communication of findings. Secrecy is the antithesis of this norm. Full and open communication is its enactment. And I would argue that data, sort of being full and open by default is what we should be striving for. Any sort of alternative is unacceptable in my view. But I'm very sort of indoctrinated by these open science values. So there's a sort of a technical problem too that I want to introduce. I discuss in the SciDataFlow paper. And this is really, there's some sort of social problems, but there's also technical problems. And we can try to solve the technical problems with better software, which is what I'm doing here. So one of the technical problems is what I call the data code divide. How many of you use Git in your research, GitHub? So pretty much everyone uses Git or GitHub. As you know, you can host your code, which is great for reproducibility on Git and GitHub. But the second you try to upload a file larger than 50 megabytes, you get a warning saying you can't do this, this isn't allowed, blah, blah, blah. And there are various extensions, but they don't typically work well with the data repositories that science uses. So I call this the data code divide or the bifurcation, where you have all this code, and what does the code do? It takes in input data, which could be sort of raw sequencing data to sets that are very large, or some sort of post-processed data sets that are sort of reduced to their size. And it analyzes that and then spits out supplementary tables, figures, other output data. And the second you put it on GitHub, it breaks these things apart. And so how do we link these two things together? The code will not work without the input data. And then the sort of data that is produced by the code is separated from it. So you can't even really see what's going on underneath the hood. There's also this other problem that I've encountered a lot. I'm sure you all have kind of encountered this, which is that you have some really complicated pipeline, and you have some sort of script, and you change the script version here, you make some subtle change, and you sort of say, okay, well, this shouldn't affect anything downstream. You don't actually look to see whether the file has changed. How would you look? I'm curious to people. How would you do this? How many people use SnakeMake? The majority of people maybe use SnakeMake. SnakeMake sort of solves this problem because it looks at the checksum values of the output data and the modification times. And if it changes, it knows to update the rest of the pipeline and rerun all these steps. But a lot of times, you can imagine you're using BCF tools X, and then the sort of cluster updates to Y, fixes a bug, changes this data, but you don't necessarily know to rerun all of this part, and your final results are invalid, so to speak, in software terms. So this is another big sort of technical problem that I've seen happen. So what does SciDataFlow do to sort of alleviate these problems? So should sort of give sort of more introduction here. So I wrote Bioinformatics Data Skill. This isn't a plug. Don't buy it. Just go to the library, or I'll give you a PDF. But and I figured when I wrote this, I was a very optimistic young kid, I guess, and I was like, 3 to 5 years, this is going to be a solved problem. Like, we're just going to have some software that links Git and GitHub and the data, and NIH is going to fund this, and it's just going to be reproducibility is going to be super easy. We're going to be able to have like a Docker container and click this and find an error in someone's analysis. Oh, wow, science is moving forward. It's not this way at all. It doesn't work this way. It's so annoying and tragic, and it irritates me. So again, more of why I decided to write this. So the actual story, I always like hearing stories about how things come about. So I went to a conference in Italy with Will, and I was stuck in my hotel room because after a really nice, like 30 hours in Italy, I got COVID. And I spent a whole day, probably more than a day, trying to organize data for an upload because I was submitting a paper and it needed to be public. And I was like, why is this so hard? This is just so stupidly hard that I have to go through and document all this stuff and say, I have this really complicated project directory and I want to make this file supplementary material and this file not supplementary material. And oh, did this change? It was just horrible. So as I mentioned earlier, I realized that this is sort of a technological problem, not just sort of a social incentives kind of problem. And so I wanted to sort of lower the barrier to make this easier and make it so that there's some piece of technology that scientists could use that they would want to use that would improve reproducibility as a byproduct. So I knew that SciDataFlow needed to be really fast, right? Because you're processing really large data sets potentially, and I'm going to be calculating checksums, which is an expensive activity. It needed to also be very robust and extremely well tested. And the idea here is that what I really didn't want to have happen is someone a year later, 2 years later, say, hey, I just lost a year's worth of work because I checked it into SciDataFlow and there's a really, rare risk corner case bug that just annihilated all of my data, right? That would be terrifying. I tell anyone, and I say this at the end as well, always back up your data. Don't trust any software or any server to not mess up all of your data. I've heard far too many horror stories of this happening. So it needed to be fast, robust, extremely well tested as well. Because yes, I'll talk about it. It's interacting with these remote repositories, which is a difficult thing to do. So I prototyped this in Python in about a day or two, and it was just really, really, really slow. So I switched to this language Rust, which I'll give sort of a random plug for. This is a shameless plug. So Rust is a language that's incredibly fast. It's as fast as C and C++, and I'm finding that it's much faster in most cases. I don't really understand why, but at least in practice, it seems to be very faster than those sort of foundational languages. It's memory safe, meaning that you will not end up with any kind of seg faults due to memory being freed when it shouldn't be freed or dereferencing a null pointer. It supports asynchronous operations. So this is really handy with SciDataFlow because you're constantly comparing different files and you're touching the file system. And the second that you touch the file system, as I'm sure you all know, it slows everything down. It's a very slow way to interact with the disk in many cases. So what you want to do is you want to sort of read something from disk, but not halt the entire program. And that's what Rust allows. It's built into the core language. You access a file, and then the rest of the program keeps running, and then it says, "Okay, I have your results for you." So I knew that once I saw that Rust had asynchronous support, that it would help SciDataFlow a lot. It's compiled, super easy to install. Again, this is really an important language, I think, for the future of computational biology. Don't learn, I would say, if you use Python and R right now, Rust is not important to learn. If you're thinking about learning C or C++, because you need faster compute times, do not learn those languages, learn Rust instead. And you can email me and I'll help you out on how to get oriented with that. I think if some people are interested, I can give a talk on that. But it solves dependency issues. Again, if you've ever used Python, it's a miserable language when you try to install something. And it's built-in testing, and it's extremely fun to write in. So you all probably know who Heng Li is. He's sort of the father of all really good bioinformatics software. It's extremely fast, written in C that is just ingenious C. I mean, I learned C by reading his code. And so he wrote this funny blog post about Rust, right? He's such a, good C programmer. He loves C, but he was like, if you really twist my arm, he said, if you hold a gun to my head and force me to recommend a new sort of high performance language, say Rust. He would say Rust. This is what I wrote SciDataFlow in. And sort of the architecture of SciDataFlow is it's a common application programming interface or API to different data repositories. So how many of you use Figshare? How many of you use Zenodo? How many have used Data Dryad? This is good because I don't support Data Dryad yet, but I probably won't. So it's good to see it's the least popular of those three. Yeah, I don't think anyone Data Dryad is going to watch this talk, but I don't feel bad about saying that. So, SciDataFlow unifies the APIs to this, and what that means is that you have a command line tool that will push and pull from these two sort of data repositories, because all of the programming side is all the complexity of dealing with these two different types of APIs is done by SciDataFlow. And yeah, so I didn't include Data Dryad because it makes it really hard to get the sort of tokens that you need to get. You have to email someone, and these other websites, you just log on and it'll give you a token easily. So what sort of underpins SciDataFlow is what I call the DataManifest. And you've probably encountered sort of similar data manifests, when you go download some large sequencing data set or something. In my case, it's the SciDataFlow data manifest is just a YAML file, so it's very easy to read. It's human readable. You can also edit it. I wouldn't recommend that because ideally it's going to be edited sort of by the SciDataFlow tool itself. But the idea is that it's just a very simple, manifest containing this is the file that you're doing this. It's tracked by this remote. This is its last checksum value, MD5, and it has some size and bytes. And then it has a remote section. Actually, there we go. Files registered with SciDataFlow. There's remote data repositories here. So this just contains the sort of minimal set of information for each particular remote. Again, you don't have to program this or write this yourself. This is just human readable stuff that SciDataFlow will generate for you. And then there's a metadata section, and this is something that I think I want to expand on because I think this is a nice feature. But really all of these files that are tracked, they should have, or at least encourage the user to have lots of metadata associated with them. So if some scientists, a year later, something comes along and they're trying to figure out what sample corresponds to what, it should be in sort of a metadata section. I'm really flying through this. Any questions so far? So now I'm just going to sort of talk about how to use SciDataFlow. So first, you can install Rust. I don't know if anyone has an interest in doing this now, but you just go to this website and it's a one single thing to install. It'll install it locally in your user home directory, which is really nice because it means that anytime you install any Rust software, you don't have to fight permissions. Everything is managed locally. And then from there, all you have to do is cargo install SciDataFlow, and it will download it and compile it on your system. I think this is actually another really strength of Rust, is that installing software is trivially easy. You don't have to download and compile everything. Yes.
Yeah, I was just wondering if we, are you gonna go through a lot of, can we follow through?
You can totally follow.
I think that would be cool.
Yeah. I'm not going to do I can do a live demonstration too. I had it worked out so I would just show you what to do. But yeah, if you want to, I can take a minute or two of people.
Thank you. Fine.
Yeah, we have tons of time. I didn't realize I was talking so quickly.
You're good.
Get some pizza. We need like music or something, some elevator music. That's okay. Is it working?
It's installing. Yeah.
Yeah, so this isn't supposed to be this much of a plug of Rust, but as you'll notice, when you install something, it'll download all the dependencies. And so one of the strengths of Rust is that SciDataFlow relies on lots of other libraries to interact with websites, to do different testing, really like framework testing for when you're interacting with a web API. And I didn't have to write all that stuff. It's just stuff that other people have written, and it's automatically downloaded. There's no fighting dependencies as there is with Python and C++ programs. It's just amazing how well this is working in practice. I wonder if said that I will certainly jinx someone in the audience. It will not work. Everyone's downloaded? Can I keep going? Okay. Okay, so the SciDataFlow command line interface is how you're going to interact with SciDataFlow. And it's designed to work basically exactly like Git. So if you've used Git, you probably kind of already know how to use SciDataFlow. This was, I think, a big part of the design for me, is I knew that it could just be really complicated if not designed right. So I just mimicked Git because I felt like it's what scientists are sort of familiar with. So there's a bunch of different subcommands. We're going to step through those right now. So first, you're going to want to configure SciDataFlow. And the way you can do this is with this command sdfconfig. And just type out --name, enter your name. And the reason why we're doing this is that I think it's Zenodo is the data repository, but they make use of, no, maybe it is Figure, actually, I'd have to check. But one of these repositories makes use of this data. Like when you try to submit new data, it needs to, it'll ask what your affiliation is, it'll ask what your email is. And I also take the SciDataFlow program takes all your email and I just sell that for money. No, I'm just joking. Thumbs up when you're all ready.
A moment, where we need to add it to our path first before doing anything.
Oh, I see. Yeah. Yeah, sometimes Russ will add it to your path automatically, or it'll give you something at the bottom that says copy and paste this, add it to your path. Yeah, it's just adding that Rust binary directory. All right. Okay, so the best way to sort of follow along would be to sort of make a new directory that mimics a project, right? So do MK, maybe I should I do a little interactive session? Should I risk it? I guess I am sharing, I'm not sharing this screen, I'll share my.
Yeah. Kind of
So source, project. So SDF in it is the first thing to do. Yeah.
Is this gonna be linked to a GitHub?
So it's completely separate from Git and GitHub, but I should have mentioned this, actually, I didn't include a slide on this. I highly recommend, in fact, it's basically imperative that you check the data manifest into Git and then post it on GitHub. And it's a small file. And the idea is if you check in the data manifest, which should live in the sort of root directory of your project, if you check that into Git and GitHub, when I clone your repository, and I want to download all those files that you may have pushed to ClickShare or Zenodo, all I have to do is SDF pull, and it will see in that data manifest all of the files, and suddenly I replicate your data environment on my machine. So that's kind of the big design of, yeah.
So ideally,
Sorry, what?
Connects the code to the data.
It connects the code to the data, exactly.
we will do this inside a Git repo.
Yeah, so actually, let's mimic it completely. Let's also do Git init.
Okay.
So now, if I look here, you can see there's two things. There's the standard sort of .git directory, and then the data manifest. And this is what an empty data manifest looks like. No files, no remotes, no metadata. Then I'll just say, if you get sort of lost with the SDF command, again, I love Rust so much, but Rust generates all this for me. It turns codes documentation into a command line application, which is incredible. Getting a nice interface like this in C or C++ is a total nightmare. So yeah, you can see all the commands. It's very much like Git. So now, let's make a fake file. I'm just going to do, maybe I'll make directory data if you want to follow along, and then vim data fake data.tsv.
Wait, when I try to SDF init, it says that I have to set SDF config.
Oh yeah, I think you didn't do
Maybe
the
something.
previous
Set.
one. Yeah.
Oh, there we go. Okay. Figure that out.
But yeah, so now I have, let's make some fake data. 3.1. Okay. 0.2. Oops. This is groundbreaking scientific data. Okay, so now what we're going to do is we're going to do SDF add. And what this is going to do is add this particular data file into the data manifest. And so the second that we do that, it'll say it added one file. Can you all see this? Okay. And then if I do, let's actually just cat the data manifest, so you can just sort of see. So what did it do? It sort of said, let's calculate how big it is. Let's calculate the checksum. Here's the path. And the paths like Git are all relative to the project root directory. Wherever that data manifest is, that's where the project root is. But a better way to do this is you don't actually want to be working and looking at the data manifest. You want to use the SDF-based subcommands. So you would do SDF status, and this is like a little progress bar, because as you get more and more data, it'll be larger. Calculating the MD5 checksums actually takes a little bit of time. It's done in parallel, which is kind of nice. And you can see that there's one thing in the data directory. So it's organized by directory. It's fake data here. It hasn't changed since the last time we added it. And here's the end of the hash, or beginning of the hash, modification date and time. So now what I'll say, so what was the second problem I talked about? It was the silent data modification issue. You have some really complicated pipeline, you rerun something and you want to make sure that it didn't sort of impact your results. How would you do that? So let's pretend, for example, that I rerun my pipeline and it changes 1.2 to 1.21, just, huge, messed up everything. If I run SDF status now, it'll sort of say, hey, something changed. What was in the repository previously that had this hash now has this hash. And you can see the status has changed. It's red. It's warning you, saying, look out, things have happened. So what if I want to sort of say, okay, these are changes that are good. I want to now cache this modified version's checksum in the data manifest. SDF update is how you would do that. Data. Do that. Type status. You should see now that it is current again, and the checksum has been modified. And this is all stuff about not having a remote setup. Where are we on? This. That. Okay, adding and moving files. So you can just do, let's say if I did touch data, data. Yeah, add.
What does the touch command do?
Oh, touch just creates an empty file.
Ah, okay.
Yeah. So you can see tree is, you guys ever use tree? Tree is like my favorite command. It makes little trees. So you can see the organization of your directories. So you can see now that there's crap data in there. If I do SDF status, you can see now I have both in there. And if I do SDF remove data, we'll remove it. This, and I will say it is still there. SciDataFlow by default will not remove the actual file. It's just removing it from this. Again, I'm very nervous about messing up people's data. I think, let's see, I don't even remember if I have an option. I can add in a delete option if people find that helpful. Yeah, no, it just removes it from there. So you would then have to rm-rf or whatever the file to remove it from your file system. But now it's out of the data manifest. You can also move things. So if I did SDF move data, real data, TSV. You notice SDF is very quick to type. It was also by design. There you go. It's been renamed in this file. So the next bit, the next bit's a little bit harder to do. I mean, let's see. You would have to create a token on Figshare or Zenodo and do all that stuff. So I think at this point, I'm going to transition to this and just show it. You see that on there? Okay, so how do you connect it with a remote? This is sort of the last part of SciDataFlow. Again, this is not a complicated program. It's horribly complicated code-wise. Like again, anytime interacting with Figshare and Zenodo, and I unit tested that extensively. So there's like thousands of lines of code that basically build up a fake version of Figshare and Zenodo and run it on my system so I can check that the API calls are working. Like the engineering behind the hood is horribly complicated just to get like a very simple thing that's fairly easy to use here. But so, yeah, that's just to make sure it's working correctly and there's no sort of software progression. But so how do you connect your local sort of repositories data, in this case, data directory, with the remote repository? So the idea is that a single directory on your system is going to be linked to some remote data repository. And so Figshare and Zenodo are not designed to have a nested directory structure. They have a single sort of directory handle data. So you will not be able to sort of have, you know, a whole complicated nested directory structure mirrored on these services. The idea is, I would say the recommended workflow is create a data / supplementary data, and then put the things that you want to share in there, and then link that to a remote repository. I'm seeing some slightly confused faces. Does that make sense, or any questions? Yeah.
So is this something that you do like at the end of your project when you're publishing, rather than like something you'd kind of track your project throughout?
Great question. It's totally up to you. I think I do it. I'd probably do it in the later part, but there are SciDataFlow users that do it very early on. They actually will create a private sort of data repository on, let's say, Zenodo, and they will upload their files and treat that like a backup, right? Which is maybe not the best backup, but yeah, well, that's a different issue. But they will immediately, as data files are being created, start pushing them to the remotes. There are file size limitations on these websites as well. I've had colleagues bump into those limitations. I think Zenodo is fairly large. It's hundreds of gigabytes, but I'd have to check. But there's also, you can get around these limitations sometimes by having multiple Zenodo repositories for a single project. That's a little bit sneaky. And actually SciDataFlow's design is to kind of allow for that as well, because you can change, data to data supplementary 1, data supplementary 2, and then have 2 different Zenodo repositories for each of those, and put those links in your paper together and say, like, these are where you would find these 2 bits of data. So what is this command doing? All this is doing is it's going to add a remote to that data manifest, and it's going to link this data directory to Zenodo. And this is the token that you'll be given by these websites. And then this is the sort of remote name for the sort of data repository. So this is what it would look like. And then I also, in designing SciDataFlow, I've separated files that have been added to the manifest to files that should be tracked by the remote. And the idea of separating these 2 things is, suppose you have a very large file in a directory, and you don't want to necessarily upload that every time to the remote when you push or pull to the remote. So that file will not be pushed and pulled to the remote by default unless you sort of say track this file, which means add it to the remote. You want to sync it with the remote. So if I had some data here called population sizes and I did SDF track, it would add that to, it would say tracked equals true, which if you saw the data manifest earlier, there's a little entry for that. And then you would see if you did SDF status with remotes, you want to add this now, it would sort of say, number 1, data is going to Zenodo. It's linked this data directory to the Zenodo remote repository. And then it would sort of say the file is current, meaning it hasn't changed on this system. And it is tracked, meaning that it will be sort of synchronized with the remote. Any questions on that? And then again, if you've used Git, it's fairly simple. It's just going to be SDF push, and it will upload this file for you. So again, I don't know if you've ever used Zenodo or Figshare when uploading like a lot of supplementary files at the end of a project. Have you ever had to who's done that before? Well, it's miserable. 1, 2 people. It's miserable, right? Because you have to go to their little website, log in, drag things off your desktop, which is never where the files live. Like, who keeps, large data files on their desktop for the most part? It's usually on some cluster or some server. Here, you can just install SciDataFlow on that clustered server, add it, push it, done. You're done. It works a lot like Git. So pulling files, again, you can sort of, this is an example of a workflow where Barbara McClintock has a GitHub repository called Maze Liftover, and I CD into her, I clone her repository and I CD in there. And I say, hey, are there any remote repositories? And the idea is that if she's checked in a data manifest here, it will tell me all the data that I can download for this repository through SciDataFlow. And indeed, even though there's no data locally on this machine yet, I can sort of say, oh, there is a data directory, it's linked to Zenodo, and there's 2 files here that I want to download locally. And to do that, it's as simple as SDF pull, and it will pull those files into your local repository. And if you rerun it again, it'll sort of compare the checksums of the remote version and your local version, and it'll say, hey, these are different or these are the same. If they're the same, it will not download anything. If they're different, it'll ask you what to do. Yeah.
So just to reiterate to see if I understand this correctly, so as long as there's a manifest file in a directory, it doesn't need to be like downloaded from GitHub, but as long as I do SDF pull, like the data file get downloaded to the directory, right?
Exactly. Yeah, exactly. So imagine I was working on some project, we're collaborating, right? And you haven't even, we haven't even put it on Git yet. And you're like, oh, can you make an SDF file for me so I can get all the data? I'd say, sure thing. And I could email it to you. You drop it in that directory, SDF pull, you have those files. So, I mean, so the larger vision of SciDataFlow is that this is how we should be doing science, right? That you sort of have some sort of curated set of data files. Like my larger vision is that scientists, I mean, a lot of us I know are from Priya's lab, you work in human genomics. It's frustrating how hard it is to get these files. You're just like doing all this kind of work where you're like, okay, where is that recombination map? You go to someone's website, you're like, oh, there it is. All right, download that thing. Oh, I'll put the download command in SnakeMake. Like all of that's fine. That's how I used to do it. But the idea is that if you had a really curated manifest file that's like all essential annotation for the human genome, maybe a different manifest file for all the BAMs that you need to get, you can just pull that into your repository. And there's a standardized set of data. There's like an analysis set of data that's curated in one of these manifests, and anyone can sort of work with it immediately. Because that's how it should work. Why we live in the 21st century. It shouldn't just be clicking and copying and pasting, like bad way to do it. Yeah.
I just want to say you had me questioning reality when you said Barbara McClintock had a GitHub.
Yeah. Sorry, yeah.
I was like, sorry, what?
Tragically, yeah. In my book, I put lots of silly names like this as Easter eggs. I don't know. Pardon my silly inside jokes. Any other questions? So we're pretty much done. Oh, and this is one of my favorite things. So you all have probably needed to download lots of files for some project. Again, human genomics is kind of a classic example. You have maybe hundreds of BAM files that you have links for, and you want to just sort of download them in bulk. Actually, do I have an example of this? I have this. Oh, I do have it. Let me. So this is what these files look like. This is a very simple example, but here you can see there's just some annotation files. And it's labeled, like, I think it's good that they have headers and there's other metadata here. But SciDataFlow has a command. So if I were to say, make directory data, human annotation, CD data, annotation, and then I'm going to move a file, this directory like this. So now you can see that there's that TSV file. I'm going to use this bulk command to download all those. And it's a very simple command. You can sort of indicate which column has the URLs in it. You indicate whether there's a header that it should ignore, and then whether to overwrite the files. And with that, you can just do SDF bulk, column 2, header. Column 2, header, that, and then I'm just going to use this file. You can see it will download with these nice little messages in parallel. Rust. Yeah, and it'll just crank through. It'll download things as fast as possible. It's all asynchronous. Maybe I shouldn't download this giant file from my desktop, which is running out of disk space. But anyways, yeah, so it'll download everything. There's also, I didn't mention this in the talk. Cancel this for a second. You see that none of these have actually downloaded all the way, which is why they haven't been put in this repository yet. But let's see. think that's pretty much the end of the talk, but I want to mention one other thing, which is that I created something called SciDataFlow assets, and the idea is that there should be some sort of community curation of these data manifest files for common projects. Again, I'm thinking about human genomics, where I'm now working in. There shouldn't be, at least in my view, there should be a lot of different ways to organize a project directory's critical files, right? There should just be some data HG38 recombination map directory or some, GFF directory for different annotation files. It should be well organized. So the idea is that we as a community, or even as a lab, even if it's labs specific, that's totally fine too. You can create your own sort of directory structure, and then put the data where it would be most well organized, and then create a data manifest, and you can host that sort of what I call an asset file, which all it is just the data manifest. And then you would just do SciDataFlow asset, and you would send it a link, and it would download the data manifest, as we were talking about earlier, in your directory, and then you can do SDF pull, and it will bring in all that data in a split second. So if your lab is, everyone in your lab is sort of, using very similar project files, like, these annotation files, agree on a structure, on a directory structure, make a SciDataFlow data manifest for that, organize it, and then you will save everyone else in the lab for the next many, many years the trouble of having to do this again and again. That's pretty much all I have. SciDataFlow is not a backup tool. Keep backups, don't mess around. And it does not manage different versions of files, because the only thing that SciDataFlow keeps is that data manifest. So if you overwrite some critical data file, it will not be able to go back unless you backed it up some other way. So all of this is to say is backup is different than SciDataFlow. Separate those things in your head. Yeah, and then thanks to these folks for various Russ advice, and to Rasmus for supporting me in this project. I sort of I came to Rasmus and I was like, I really want to just write this thing. I think it'll take me not that long. He's very supportive, and he cares about, open data as well. So I think it's nice to get the opportunity to bash this little side project out. So with that, I'm happy to take any questions. Yeah.
This is more of a, your own like trajectory towards kind of this work. Yeah, I guess what has been your experience writing packages for bioinformatics and all of this?
Well, that's a good question.
You mentioned how much crazy engineering there is behind making it look so like smooth and sleek.
Yeah, it's just, I'm tempted to almost just show you.
Sure.
So a lot of this is just gibberish. Yeah, so it's not too complicated. You can see I have Dryad in here, but then I refuse to continue development on that. I think it's like a lot of the logic is this project file, which is figuring out where things are in your directory. It's also all of these APIs, these web APIs. This is what I was mentioning earlier. That's incredibly complicated. All of these web APIs are something that you can interact with through the SciDataFlow library itself. So you can actually, if you want to develop your own Rust program, but interact with data on these repositories differently, you can use the logic that I've written. So it's organized to not just be a command line tool, but also a library for the more advanced users that want to do their own thing. So you can do that. The other thing I'll just say is I kept talking about testing. I don't know if this was making a lot of sense. Rust is such an important language because it has integrated testing frameworks. Python does not have an integrated testing framework, or they do, but no one really uses it because it's crappy. So this is a language that treats testing code, writing code that tests other code, as not just an important part of the language, but a critical part of the language. It's just so cool. So all of the Rust commands are based on this program cargo. So in my Rust directory, if I did cargo test, well, actually I have to do one thing. One trick, I have to do single threading, otherwise the test will fail. History. This is where the live demo fails.
You were almost there.
It's now not working. Other dashes. So it will run through all of these tests. And as I mentioned, test whenever you're interacting with Zenodo or some remote thing, every time I run a test, I can't just be bashing Zenodo's web servers, right? And I should say that all of these tests are run by the Rust servers as well. So when I host the SciDataFlow code on Rust servers, it will test everything to make sure everything's working. So I can't just be bashing Zenodo and Figshare, their web servers, every time a test is run. So all of these tests, they build up a local web server that emulates how it works, which is, again, a horrible amount of coding that I had to do. And it will interact and test my code against those requests to interact things. It will say, hey, I want to upload this, will this work? I test various errors. So a huge amount of the sort of programming side of SciDataFlow is just making sure that this won't break or lead to a loss of data. Because again, in mission sort of critical scientific applications, is what you need to do is unit test your framework. How many people unit test code? Don't feel ashamed. Anyone? 1 to 2 people? Yeah, great. You should not feel bad about not unit testing your code because science doesn't prioritize it and it's terrible, but it's not your fault. It's that science doesn't give a shit about whether the code is working or not. But in industry, if you all go to industry jobs, they will just be like, what? You don't unit test code? Because they know that to have a product that can be reliable, you have to unit test code. That's my little spiel about unit testing. But yeah, there's no incentive to do it in science because it doesn't really matter. Except for I would say it helps if you write some core unit test for very finicky code, I recommend that. So yeah, you're asking about project, package development. It's a huge amount of work because of all this testing and having to do test-driven development. Although you kind of get in a groove, I would say, and Rust makes that easier than other languages like Python. Yeah, I find that, I guess it's hard because you can get little publications out of these things, but they're not seen as like a big contribution to science, even though I think they're very important to solve these little technological problems. So I don't know what to say. I have lots of mixed feelings about it. It's good. I mean, if you see little projects like this, or you want to contribute to SciDataFlow, definitely do them. They're helpful. And just get people riled up about the fact that you need to be sharing data more. You probably need to be testing things more. Probably need to be replicating things more. But these aren't incentivized. So some unknown fraction of our science is wrong. Even if you wanted to look, you probably wouldn't be able to find the data. Depressing. Yeah.
So I have 2 questions. So one is because when you mentioned the control version of data, that could be super cool. Like, are you thinking about like expanding into that or not really? Because that could like solve a lot of things too.
Yeah, so actually versioning the data.
Yeah, like Git,
Yeah.
but like instead of for code for data.
I think there is a need for that. It's really hard because at least with the data that I work with, the data is just so large and it's already kind of unmanageably large on most clusters that to have different versions would require it would probably double the disk space easily. And that's only for one version back, right? But as it's modified, yeah, there's other ways to do it, but it requires keeping diffs on binary files. Like you can imagine having a gzipped file, that's a simple case actually, a gzipped text file of, let's say, a VCF file. And let's say like a few of the variants got changed and you want to have a record of those things. You could, like very advanced software could look at the bit difference and they would say like at this bit, it started changing to this bit, and then it would be able to compress those differences, but it's still a giant nightmare, I think, to code that up and do.
Well, I was not thinking about raw data like VCF file, well, not VCF file, but like, I don't know, sequence data, but more like if you're writing a pipeline, sometimes I end up having like version 1 CSV, version 2 CSV to see what my pipeline changed in the result.
Yeah.
So actually having like a version control of the data, but mostly results, right? Like
Yeah.
selection coefficients or whatever. That would be really cool.
Yeah, well, so SciDataFlow will keep one version back, but it doesn't keep the version. It just sort of says it changed.
Yeah.
If you have smaller data like that, if it's less than 50 megabytes, I usually just check it into Git, because then it solves the code data bifurcation problem, and you can look at as version control of the data file itself.
Yeah. Yeah. That makes sense. And another question is, so I think something, because I really want to implement it, but like something that I wasn't sure about. So you said that Windows like Senado have like data in bulks, right? But based on the data manifesto, if you download it with your software, then it will like put it in like a tree structure. It will immediately structure the data.
It will mimic it as it was in the structure that it was created in, right? So the
Okay,
idea is it will completely sort of, recapitulate the original design of the data project directory. Which is by design, so another thing, don't code with absolute paths, like you probably all know this, but never use absolute paths in your scripts, use relative paths. And by doing that, it means that when you use SciDataFlow to bring in all your data and you rerun some script, as long as it's all relative paths, anyone can run those scripts. They should all work and it'll say, oh, this file's there, this file's here.
I was curious, I guess the primary use case, you're using SciDataFlow kind of at the end of your project. But I wonder if you've thought of use cases where it's more sort of in the loop and continuous. Like I have in mind something like viral phylogenetic, where you're sort of monitoring something and the data and the code are changing at a similar time scale. Do you think there's an opportunity for SciDataFlow to kind of, I don't know, help people doing that kind of work?
Yeah, I think it would. I mean, it won't keep multiple versions, but it'll definitely allow, well, I think in the phylogenetic world, it would just be nice to have the data hosted somewhere and the code somewhere else, and then have a single command where you can pull in the latest data, and then also push new versions of the data. So I didn't show this as a sort of an interactive example, but if I were to modify a file and I did STF status, it would sort of say, hey, this version of the file is different than the one that's on the server, and then you could use STF push and it would push the new version to the server. And again, it's very cautious. It'll make you do -- overwrite to overwrite that data, just because I want it to be very explicit to the user that, the data is going to be changed and overwritten. Yeah, I think there are a lot of use cases, and it's fun actually. So I wrote this and I expected it to be kind of used a little bit, but it's, I think, my most popular GitHub repository. Like people seem to really like it, and I get to see in the issues that people are filing. I mean, there's not a lot of bugs or anything. I get to see in the issues that file like how they're using it, and a lot of people are using it continuously like that. That's actually a much more common workflow than I anticipated. It's cool to see. So.
You also hook into SnakeMake so that if a remote file changes and you rerun your pipeline, that's just automatically detected.
Yeah, this is something I've thought about. I'd have to, I don't know how I would do that. What you could probably do is put the DataManifest in as a dependency, maybe. Would that work? I'd have to think about it more. But yeah, I do intend at some point to kind of integrate the two. The author of SnakeMake, by the way, is becoming very big into Rust. He's like a Rust guy. So I imagine that there's going to be some interesting developments. I don't know if they're going to move SnakeMake over to Rust. I can't imagine that they would, because they would need some other language to code it in other than the SnakeMake Python variant. But he is certainly a big fan of Rust, which is cool to see. So I can imagine some neat developments in the future on that front.
Okay, so we are out of time, but yeah, I think Vince might.
Yeah, I can take one more questions too.
Yeah, but in any case, I think yeah, we'll stop recording and release anybody who has to go. But thank you for coming, and thank you, Vince, for being here.
Yeah.
We can definitely keep talking about questions. Thanks.